It is my great pleasure to introduce our speaker for today. Professor Locker is the head of the research group for electron microscopy of pathogens at the Paul-Ehrlich-Institut in Langen, Germany. She received her PhD from the University of Utrecht in virology and cell biology, then worked as a postdoc at EMBL, and after stops at the University of Heidelberg and Institut Pasteur, she's now been at the Paul-Ehrlich-Institut in Langen, where her work focuses on imaging of neglected infectious diseases. Please, Jacomine, go ahead with your presentation.
Thank you for inviting me. And I was actually a little bit surprised that you invited me, because I think I'm no longer a member of Euro-BioImaging, but nonetheless, it's a great pleasure to present some of my personal views on electron microscopy here. I think the next slide I can kind of skip, because it's just an introduction that was already made by Johanna. I actually did my PhD on cell biology of coronaviruses, which in those days were not very sexy viruses, because they were just infecting mice and cats, and who cared about that, right? Well, a really exciting time was at the EMBL in Heidelberg, where I started using large DNA viruses as tools to understand cells using increasingly imaging methods, in particular electron microscopy. And based on that, I was then asked to head the EM core facility at Heidelberg University. But I made a big condition here. I said, I only do this if I'm allowed to continue my own research, because I need this to develop new techniques in electron microscopy. And they agreed, and this happened to be a very exciting time where we had really interesting collaborations with people from virology. I'll mention some of those studies later on. And then I went to Paul-Ehrlich-Institut, and I came with great hopes, because it's a big institute, very famous in Europe. Of course, focusing only on pathogens. But after 4.5 years, I found myself just doing service and administration, and this is not what I really wanted. So I was in a very fortunate position to be able to be recruited to Paul-Ehrlich-Institut and the University of Gießen, where I continue my own research working on and in particular on large DNA viruses using electron microscopy methods. Now, as most of you know, but some perhaps not, electron microscopy is faced with specific challenges. And I'm going to go through some of them later on as I'm presenting some of our data. And so basically on the left side, you see this, very basic schematics of an electron microscope, where you have an electron source, and the electrons then are in a high vacuum column going through the specimen, and then creating an image on the viewing screen at the bottom of the camera. And so this comes with two main challenges, meaning the sample has to be dry, dehydrated, and thin. And we'll see that as I go through this, there are some exceptions and techniques that have been addressing these two main challenges. But also what is really important is that sample preparation is the key. And this will be one thing that I will come back to all the time. It needs optimization depending on the sample and the question asked. And this is one of the other big challenges in electron microscopy. Now, my personal view is, and this is why I'm really at the right place in Paul-Ehrlich-Institut at the moment, is that if you want to set up electron microscopy techniques, you require a solid biological question. And if you want to acquire meaningful results, in my opinion, in electron microscopy, you need to use complementary techniques that kind of confirm each other and that can be quantified. So I'm absolutely no fan of somebody asking for a single last picture that illustrates the story of, their biochemistry results or something, or molecular biology results. So I will show you some examples as I go through various techniques in EM. And of course, there's many techniques in EM, but I would like to focus just on a few here. I would like to start off by EM immunolabeling to precisely and quantitatively localize proteins. This is what I started off with in electron microscopy. And my impression is that this technique has gone a little bit out of fashion, which is really a pity in a way. And then I would like to briefly just compare and mention two 3D EM techniques, electron tomography and focused ion beam scanning EM, and illustrate the cool things you can do with it. And then I would like to go, of course, to the big hype of today, which is cryo-EM, like to, talk a little bit very briefly where it came from and where it is now, and then finish up with some concluding remarks. So if you start with the first technique, immunolabeling in electron microscopy, here's the schematics. Is perhaps a little bit too much detail, but it starts by fixing cells, collecting them and pelleting them, infiltrating them with sucrose, and then basically flash freezing them. And then you have a pellet which is solid, which can be cut at low temperature in a cryo-microtome. And basically there's devoted microtomes that are very user-friendly these days. So you cut sections at low temperature. They are then brought to room temperature on electron microscopy grids. And then basically you can apply standard immunolabeling like you would do fluorescence microscopy. You incubate with primary antibodies and then with secondary antibodies coupled to gold particles, because obviously on an electron microscope you cannot see a fluorescent signal. You need to see something which is electron dense, like gold particles. Now, if you have your sections at the end of the labeling procedure, they're still wet, so they are not compatible with imaging. And so one thing that was actually really developed mostly by Tokiyasu, who passed away a couple of years ago, is to contrast the sections together with something which is called methyl cellulose, which is nothing more than wallpaper glue. And what this will do when it dries is to prevent the structure from collapsing. And this is essential because if you don't do this, then the structure looks really awful. Now, basically, these gold particles, as illustrated here in this first publication slide here, what you can do with them is you can basically count them and then say, okay, I have so many gold particles per membrane, or so many gold particles per such and such. And this allows you then to say precisely, where is your antigen localized. So this slide is a little bit complicated, but I will go through it systematically. This is basically HIV-1 budding into primary macrophages. And when you look at this first image, this overview image first, basically what you can see is a lot of viruses in vacuoles. And so basically a group before us suggested that these vacuoles are actually endosomes. Now, we were not quite convinced about this, and so what we did is we used two markers, one marker of endosomes and one marker of the plasma membrane, CD63 of endosomes and CD44 of the plasma membrane. And what you could do by simply counting the gold particles is to show that the membrane of these vacuoles are actually derived from the plasma membrane. And we could also show that actually the viruses themselves were labeled with this CD44. So, of course, we are not convinced by simply doing this experiment, so we did additional complementary experiments showing that if you use a membrane impermeable dye, primary human macrophages make this enormous amount of plasma membrane invaginations, and this is really where the virus buds. So rather than budding into endosomes, it buds into plasma membrane invaginations, and occasionally you have this very lucky shot where you see the viruses in this kind of sac-like thing with an opening to the outside world. So, as I said, I think that for my personal view is that EM immunolabeling, and I think that some of the students that I supervised in this technique, but also in other EM techniques would agree with me. My personal view is that EM immunolabeling is one of the most challenging techniques in EM. And that's probably why it's not so much in fashion anymore, I would guess. I guess it starts by asking questions like, is your antibody labeling really specific? I mean, what kind of control are you going to use here? If you have a cellular protein, are you going to knock down the protein to make sure that your antibody labeling is specific? And then you find out that if you do so, that some cells no longer synthesize a protein, but others still do have labeling. So what do you then say? I mean, how do you interpret this? And without failure, labeling conditions require optimization. You need to, change the blocking solution, incubation times, etc., etc. So it's quite kind of tedious. And so coming back to, knowing something about the biology, I think a very good a priori knowledge of the protein, its biology is really required. And I illustrated this by taking two random pictures, fluorescent pictures from websites, where on the left you have this single fluorescent dot. And of course, if you start labeling, then you cannot expect a lot of labeling. You can only expect some sections to be labeled, and that this in a very tiny area of the cell. And you hope that you actually find these very tiny areas of the cell. And as opposed to the image on the right side, you have almost labeling everywhere, even though these are integrins. And so, but you expect perhaps a labeling that is a little bit more concentrated at the plasma membrane, but then you would expect also labeling in the cytoplasm. So coming back to this idea of the fact that the sample has to be thin. So this, of course, is a big challenge in a way, because most of us are probably working with cells, let's say HeLa cells that grow in Petri dishes. And a HeLa cell has an average thickness of about 10 micrometers, but we are working with sections that have only 50 to 70 nanometers. So you lose one dimension. And illustrated here with this orange, you cannot look into the entire orange because it's too thick. You're just seeing a slice. So I would like to basically present here two possible complementary solutions. There's more in the field. And just to show you, what you can do with them if you use them in parallel. It's exciting sometimes. So on the one hand is electron tomography, which is based on transmission EM. And it's usually a transmission EM that operates at a slightly higher voltage. So you can use slightly thicker sections. And then what is special about this microscope is that the stage can be tilted and 2D projections automatically recorded, resulting in multiple 2D projections that can then be aligned into a 3D model. Now, the advantage of this method is that you have detail, resolution is good, the acquisition doesn't take so long, perhaps an hour or so if your microscope is well aligned. That means that you can make many tomograms in one day, especially if you're a PhD student and you work more than 8 hours a day. Perhaps you can also, program your microscope that it makes the tomograms automatically for you, also possible. So you can get a lot of quantitative data, many tomograms of the same kind of structures. As opposed to this, there's focused ion beam SEM, but again, of course, here, what is really limiting is the section thickness. We were talking about cells of 10 micrometers, but the section thickness is maximum 200 to 300 nanometers. So you still don't have the entire volume of a cell. So as opposed to this, you have focused ion beam SEM, which would be typically to generate a 3-dimensional acquisition of a whole cell. So how does this work? You have SEM imaging, so scanning EM. So you basically image the surface. Then you have an additional ion beam, which will cut a thin slice of the surface. And then by repeating imaging and slicing, you'll get a 3D impression of a whole cell. Now, the resolution is usually not as good because it's, SEM. Used to be a very big disadvantage used to be that it takes very long. If you want to have a good resolution and go through an entire cell, it used to take, like with this Auriga here on the right side, like 2 or 3 days. Apparently the new machines are a little bit faster because they have a different way of acquiring images. But still, it takes overnight, and then you find out you chose the wrong cell. So then you have to repeat from scratch. So you can imagine that this technique is not as quantitative. Nonetheless, I would like to show you what you can do with it if you compare. This is from my own work where we studied the membrane acquisition of a large DNA virus, mollivirus. So on the left side, you have a tomogram where you can see details on how the membrane is organized. What you do not see very well in this tomogram is that these membranes are actually acquired by small 50 nanometer vesicles. And what you can see here on the right side is the corresponding focused ion beam SEM of a similar infected cell, where now you have an entire overview over the entire cell. The yellow things here are the mitochondria, which are pushed away from the viral factory. The viruses are here in red, and what you can see here in 3D very nicely is how the viruses actually are surrounding these green stuff, which are the little vesicles that make up the viral membrane. So I'd like to show you another really nice example of a collaboration where basically we used almost exclusively electron microscopy to make up a really interesting story related to dengue virus. This is a collaboration with the group of Ralf Bautenschlager in Heidelberg some time ago, more than 10 years ago, in fact. And if you look at a thin section of a classically embedded cell, you see all of these details already. You see in the middle these spaghetti-like convoluted membranes, these vesicles, they are quite big and prominent. And then on the sides, you can see these really little dark spots indicated here with VI, which are the viruses that are arranged in really nice arrays in the endoplasmic reticulum, the lumen of the endoplasmic reticulum. Now, we did many experiments, but one of the most illustrative one was using electron tomography. So what we could show with electron tomography here is that these so-called vesicles are not closed. In fact, they have a little pore which connects them to the cytosol. So basically, they are invaginations of the endoplasmic reticulum. And basically what we could see is that opposite this pore, we always saw little viruses budding into the endoplasmic reticulum. So we came up with this model, which the reviewers apparently liked very much, where the vesicles are invagination of the endoplasmic reticulum, where genome replication takes place. The genome is then extruded through this pore into the cytoplasm, and then taken up immediately by budding virus particles into the ER lumen. And in fact, for all flaviviruses, because dengue virus is a member of the flaviviruses, this model was actually confirmed. So it seems that flaviviruses are all doing the same thing. So here is my personal intermezzo. I think that the biological question really pushes the choice of the methods and its optimization, and not the other way around. But let's look at the big hype on cryo-EM that's going on at the moment. Now, I told you that classically samples are dehydrated and chemically fixed, and biological samples obviously contain a lot of water. So that's not so good. And basically what cryo-EM is doing is it's preserving cells by rapid freezing rather than by chemically fixing. And so the sample can be imaged by a cryo-EM, by dedicated cryo transmission EMs in a fully hydrated and frozen state. And so it produces presumably images that are considered to be close to native. Now, we take this technique more or less for granted, but I would like to take one step back here, because I was in a very fortunate position to be able to attend a lecture from Jacques Dubochet, who obtained the Nobel Prize with two other colleagues for cryo-EM, in fact. And he was the one who was actually doubting whether you could freeze water so fast that you don't make ice crystals. Because as you know, if you freeze something in a -20 and then you thaw it, you have a lot of water coming out of your sample, because your vegetables are basically destroyed by the ice crystals in your vegetables. So he was doubted this, but he took him a year basically to realize that if you freeze fast enough, then you can actually generate vitreous ice, so glass-like ice and not hexagonal ice. We all take this for granted, but it took really, big minds to come and realize this. Now, on paper, this experiment, preparing samples for cryo-EM, is really simple. You have an electron microscopy grid, usually a devoted electron microscopy grid with holes in them and a little carbon layer. And you pipette your sample in a little drop on the electron microscopy grid. You blot away excess water, liquid, and then you plunge freeze it in liquid nitrogen cooled ethane. Why ethane and not liquid nitrogen? That's because ethane, when it's cooled down, it freezes much faster than liquid nitrogen. If you would basically, those of you who are sometimes snap freezing samples, in liquid nitrogen, you have this kind of boiling effect, which is actually not good. So you would have like ice on the surface of your sample. Now on the bottom, there's the, the basic setup, the manual setup. These days you have devoted machines for this. You can program the blotting time, the temperature, etc., etc. But both of them end up with a sample where basically the sample is embedded in a thin layer of vitrified ice, vitrified water, and this allows you then to image them by cryo-EM. So these samples that are now vitrified in this thin layer of ice allows you to obtain structural information, for instance, of particles, but also in the cell. I'll come back to this in a second. Again, this is from two studies that we did. On the left side, this is, of course, a SARS-CoV-2 with these really beautiful spikes around them. They were actually chemically fixed for freezing because, of course, we were not allowed to put them in a microscope without fixing. And on the right side, there's a study where we looked at tunneling nanotubes connecting various cells where cryo-EM allowed us to make conclusions about the way actin is organized within these tunneling nanotubes. Now, modern cryo-EM is now a complementary structural biology method, but it didn't used to be like this. And the resolution has actually increased tremendously in the past 10 years or so. And why is this? This was not like this in the previous century. It's mostly because of much improved hardware and software. And in particular, new cameras that detect electrons directly rather than converting them into photons. If you want to know more about this, there is a really beautiful, nicely written review about this, 2 pages long by Rainer Grolbrand in Science from 2014. So since you can now obtain almost close to atomic resolution in a cryo-electron microscope, of course, now you have big advantages over X-ray crystallography. You don't need to crystallize your protein. Some proteins don't want to crystallize altogether. So you just need to freeze them in a way. And of course, you can go look at protein complexes in their native context within the cell. And you cannot do this with X-ray crystallography. However, if you want to focus in situ protein complexes in cells, your sample should still be thin because we're still talking about transmission EM. Preferentially, if you want to have really high resolution, you tend to have a layer of ice that is no thicker than 100, perhaps 200 nanometers. And cells, of course, remember, they're like 10 micrometers thick. So there's, of course, thinning methods around. And I like to briefly mention the one that has gone out of fashion, which is cryo-EM of vitreous sections. So here you freeze your sample, your cells, by high pressure freezing or by plunge freezing. And then you use the same kind of microtome that you use for EM immunolabeling. And you cut the sections and place them at low temperature on electron microscopy grids and then observe them by cryo-EM. Now you can already see in this beautiful image in A, the big problem with this technique. You have beautifully nice sections that look like glass, but they won't lie flat on the electron microscopy grid. This is an enormous challenge. And I don't think that this has been solved properly so far. And this is a big challenge for electron tomography because electron tomography requires flat samples. So it's gone out of fashion. It's not so difficult to learn. In fact, I had a technician learning it just, in 1 week. And instead of this, now we have a dedicated machine called a cryo-FIB milling or a cryo-SAM, in fact, which uses the same idea as focused ion beam SAM. It has an SAM beam, and then it uses the focused ion beam to slice the sample. So you have cells grown on electron microscopy grids, plunge frozen, and then you thin the cell such that it's no thicker than like 100 nanometers or so. And this can then be used for electron tomography or for cryo-EM. So this has become actually more and more routine because the machines become more and more easy to handle, even though they're still very expensive. Now, these are some of the challenges in cryo-EM, and I'll give you an example in a second. Again, we come to this tedious point of making the sample. This can be so unbelievably time-consuming and tedious. And because we aim to have this sample in these holes of the electron microscopy grids, and with the eyes not too thick, not too thin, etc. I can give you an example where we had protein complexes that my negative stain looked perfect. They looked really beautiful. And then you try to freeze them, and then the sample aggregates. It doesn't want to be in the holes. The ice is too thick, the ice is too thin, and then you end up working for 6 months before you finally have the perfect sample. Now, the electron microscopes actually are becoming less and less really the bottleneck related to time. Here is this box, this cryos that people probably recognize. This is how microscopes look these days. There's the box. It's a little bit, disappointing in a way. And the imaging acquisition has become semi-automated. And if you have a good workflow in place, the imaging per se is not all that time-consuming anymore. And what do I mean by semi-automated? By semi-automated, I mean the microscope will make, of course, the tomograms and the images automatically, but you need to program it. And what you need to program is basically things like make tomograms of this area, this area, this area, because I, as an experimenter, decide that this is the best place for tomography. The best here, this is where the sample looks best. And then, of course, you need to program the parameters for imaging. Let's say, which condenser are you going to use? What kind of magnification? How many electrons are you going to use before the sample starts to burn, etc. So this is not automated. You, as an experimenter, has to do this, preferentially perhaps together with the operator of the microscope. So there needs to be, let's say, very good communication here. And then finally, you end up with tons and tons of data which needs to be processed. And this can be extremely time-consuming. I'll give you an example from this tomogram here at the bottom, which again you recognize as SARS-CoV-2. So when we did this study in the first lockdown in 2020, we were in a very fortunate position to be able to use a protocol that was developed for a other coronavirus, which worked for us straight away. So we were able to purify and concentrate the viruses from the supernatant of infected cells using roughly 2 or 3 weeks. This is really exceptionally fast. And then the eyes happened to be really perfect. And so it took the operator of the microscope 2 days to find the nice areas, and then the microscope did everything by itself. But the really time-consuming part here, and I'm talking about several months, was to analyze the structure of the spike protein of this virus. This took really 2 or 3 months. In the end, we got a really nice paper out of it. And of course, cryo-EM is now a very powerful structural biology method. And rather than saying that it's complementary to X-ray crystallography, I like to say that it can be complemented with various other methods. And I just listed here 2 of them, again, using some of the examples that we published ourselves. Now, what is very helpful if you do in situ, let's say in the cell, you see a structure and you have an idea what it could be. What is very helpful is if the X-ray structure is known. So on the right side, this is basically sections of cells vitrified. And we are looking here at the scaffold protein of this big DNA virus, vaccinia virus. And we know that in cells it makes hexamers, trimers of hexamers. So it first makes trimers and then these form hexamers. And because the X-ray structure of the trimer is known, we could actually identify it in the section. Now, of course, this is not very exciting if you look at wild-type infection, but it was very helpful when we were looking at mutant conditions, where we couldn't really directly identify the scaffold. And what we could show is that by pasting in, by basically using the X-ray structure, that the trimer was really formed under these mutant conditions, but the hexamers failed to be formed in a proper way. Now, of course, you also have prediction programs. In the case of SARS-CoV-2, what was very helpful is to combine it with molecular dynamic simulation. What I mean by that is basically when we were looking at the first images of this virus, we saw that the head here was connected to the membrane by this slender stalk. This was known. But the stalk always seemed to be somehow bended. And my first hinge was that this is a preparation artifact. You know, perhaps we blotted a little bit too much, or basically we were not careful enough by preparing the virus, etc. But what was very handy here was the idea of using molecular dynamic simulation, which is a computer-based program which can actually predict, for instance, if regions in the cell based on amino acid sequence are flexible or are changing their conformation when you bind antibodies to them, etc. And what the molecular dynamic simulation showed is that indeed the stalk of this spike is flexible, and this is mediated via 3 hinge domains that allows the stalk basically to bend over different degrees. So then we were quite confident that what we were seeing in cryo-EM is really true. Now, what is sometimes also very helpful, of course, is that if you have a structure in a cell and you have an idea what it could be, but you're not completely sure, and because again, remember, you cannot do immunolabeling here because everything is in a frozen state. But you have an idea it could be this protein, or perhaps this protein, then you can, what can be very useful if you can predict the 3D structure from amino acid sequence, and what is becoming more and more, let's say, of a hype is to use this program which is called AlphaFold, which can quite well predict the 3D structure based on amino acid sequence of proteins. This is something we are experimenting with ourselves, and we have had some luck, which we hope to publish soon this year. So I like to finish with a conclusion and then show you 2 more slides before I'm finished. So I think the take-home message that I would like to put across is that despite all this improved hardware and software, sample preparation remains the key also in cryo-EM. And cryo-EM is very powerful, but there is still in electron microscopy, generally in imaging samples in electron microscopy, there's a couple of challenges that remain. And for one thing, there is, and I didn't really mention this at all during my talk, otherwise we'll be here until tomorrow morning, I guess, which is the imaging of tissue. You know, sometimes the sample tissue, let's say you have a piece of lung from a mouse and you want to see where is SARS-CoV-2 in this lung. Sometimes it's too thick for proper freezing, only the outsides are frozen properly. This is a challenge, and of course it's way too thick to analyze efficiently in 3D. You would have to make serial sections and then do array tomography. There is methods that address this, but they are mostly based on classical room temperature electron microscopy. And of course, the other big problem in cryo-EM is that it cannot be combined with EM immunolabeling. So you cannot really identify your protein by antibody labeling. And I like to finish off with 2 slides based on something we've been experimenting with when I was still in Paul-Ehrlich-Institut, where we basically used a hybrid method. And it's based on a technique which was developed before, but published in quite some detail by Bram Koster in Leiden, where they first followed a routine protocol for EM immunolabeling. Remember this Tokuyasu immunolabeling protocol where cells are fixed, infiltrated with sucrose, and then they're flash frozen. You cut sections at low temperature, and then you transfer them to room temperature for immunolabeling. Now, the routine method will then basically dry the section and contrast it with ionoacetate. But what they showed is that what you can do instead is to freeze them again and then to analyze them by cryo-EM or cryotomography. And we've been experimenting with this a little bit. And of course, then you can identify with antibodies where is my structure of interest. Again, of course, this is from our own work. I think it's quite beautiful. Perhaps some of you don't see the details because it's cryo-EM. This, of course, is a pox virus. And you can see the bilayer of the membrane. You can see the scaffold on top of this membrane. And basically, we pasted in the X-ray structure of this scaffold protein and could come up with a model which is not refined yet. It's not finished because we never published this. Is that what we could show in addition to the scaffold is that it's somehow connected to the membrane, something that we never were able to visualize in any other method. And it would be interesting, of course, to identify this structure that mediates the binding of this scaffold protein to the viral membrane. What you see here as a blob on the right side is this striated stuff is actually the viral DNA. So I like to finish by thanking all of the numerous PhD students and postdocs that contributed to these studies that I mentioned in my talk. And the sponsors, the University of Gießen, the Cluster of Excellence from the state of Hessen, Leuven, in particular Druid, for which I am part of, Paul-Ehrlich-Institut, as well as the Deutsche Forschungsgemeinschaft. And this picture is here to emphasize that Langen is kind of a little bit of a boring place, but the institute is actually embedded in a very nice garden where we prefer to sit when we discuss our experiments. And with this, I'm actually open for questions. Do you want me to unshare or